All right, so this is how we make the mitosis flippy notes. You just need 2 pieces of paper, place one on the table, and then put the other one on top of it, but just a little bit lower, about a finger's width lower, about 2 centimeters. Then you take both of the pieces of paper and fold it upwards, again leaving about a finger's width between the 2 folds. And then what you end up with is notes that have 4 different flaps. Your next step is to take the top flap and cut it in half, and then cut each of those flaps in half. If you want to measure them, you are welcome to do so, or if you want to cut them straight, unlike my first cut there, you are welcome to do that as well. Now that we have the basis of our notes, it's time to put in the labels. Our first one is prophase. Second one is metaphase. So I'm writing these underneath the flaps here. Anaphase and telophase. And then on this flap, we're going to have cytokinesis and interphase. And finally down here, why do cells divide? All right, so what I'm going to do now is show you a finished version of these, and what you can do is pause this video in each section and then copy it down onto your own notes. So here's the top. You have the first phase is prophase. Spindle fibers form. The long and thin chromatin condense to make these little chromatids. Now, this right here is one DNA molecule, and this is one identical DNA molecule because it was replicated in order to make 2 new cells. So here's one DNA molecule, here's an identical DNA molecule. And so they condense up to make it easier to separate them out. The membrane around the nucleus disappears. Okay? And like I said, these little spindle fibers form, and their significance is going to become clear after this. So there's that picture, and then here is a description of what happens. You can pause it here and copy that down. Our next phase is metaphase. Now, the 2 chromosomes, and by the way, we're doing an ant cell because ants only have 2 chromosomes. I don't want to do a human with 46. That'd be too much. So the 2 chromosomes line up in the middle. This is metaphase. A good way to remember is meta starts with M and middle starts with M. And the spindle fibers go and attach to these little structures called centromeres. Spindle fiber comes, attaches to the centromeres. I'm going to flip it. You can pause and copy. Our next phase is anaphase, where the sister chromatids are being pulled apart because you're going to have 2 new cells, one here and one here. And so they need to separate. And then finally, we have telophase. In telophase, 2 new nuclei form. So the membrane comes back around both of them. So it's like we have 2 cells in one. So finally, they need to pinch apart. Nuclei is plural for nucleus. Now we have cytokinesis. Cytokinesis is when the 2 new nuclei are pinched apart. Now in animal cells, they're pinched apart because they're more flexible, but in plant cells, they have this rigid cell wall around them, and so it has to build a new cell wall in between, so it's a little bit different. Now, interphase is where the cell spends most of its time. So this is actually only 2 DNA molecules, the blue one and the red one, but they're all stretched out and elongated. And when they're stretched out and elongated, they're called chromatin. And so this is the way a cell is most of its life. When it's not reproducing, it's like this. And what does it do during that time? Well, it just does its normal stuff, like making proteins. For instance, if it's a pancreas cell, it's going to make insulin and do all of its jobs. If it's a skin cell, it's going to protect you. When it's about to divide, then the DNA replicates. So that happens during interphase. Why do cells divide? To grow, to replace old and damaged cells, and for asexual reproduction. A species, like you might remember from our lab, we saw hydras in the pond water. They had these like tentacles, and they were really cool. Well, a way that they can reproduce is called budding. And it's where they grow a whole new little dude that's identical to them. So they use this process of cell division because this process makes two cells that are totally identical to the original cell. And that's what's going on right here. And when it goes big enough, it drops off and becomes a whole new hydra. So occasionally, asexual reproduction is the reason. So this kind of cell division does not produce sperm or eggs. That is a different process called meiosis. And it's different because a sperm and an egg only has half of the DNA, whereas in this case, the final result has a full set of DNA. All right, remember, you can pause at any point in this video if you need to copy stuff down. Thank you.